Hello, I'm Dr. Michael McInnes. I'm an internist, and the lecture today is on drug receptor interactions and pharmacodynamics. So in this lecture, we're going to be discussing how drugs interact with their receptors to exert their effects. Most drugs do use some type of receptor to exert their effects on the body, and these receptors can either be extracellular, outside the cell, stuck onto the cell membrane, or they can be intracellular receptors. The drug effects come about in various different ways. Sometimes drugs can bind to specific enzymes and inhibit or suppress or stimulate their catalytic action. Sometimes drugs can bind to nucleic acids and inhibit gene transcription. Sometimes drugs can bind to receptors on the membrane and open an ion channel. And in all of these cases, it's the drug-receptor complex, the joining together of the two, that leads to some type of biological response. Most cells have lots and lots of different receptors, but those receptors are specific for certain ligands or certain drugs. Now, you'll hear that word ligand a lot. Sometimes people will pronounce it ligand or ligand, but I looked it up, and ligand is appropriate, so that's what I'm going to say. So a ligand is anything that binds to a receptor or an enzyme. It could be a neurotransmitter, such as epinephrine. It could be other endogenous chemicals, such as histamine. It could be a drug. It could be an enzyme substrate. So a ligand can be almost anything. And when a drug or a ligand binds a receptor, it causes a conformational change in that receptor. And that change in the receptor leads to a biochemical effect. As I said, it may open an ion channel and allow ions to flow into the cell membrane or across the cell membrane and into the cell, and that can affect membrane polarity. Or it could change the receptor's conformation so that it opens up a binding site in an enzymatic portion of that receptor, so that enzyme is now available to perform its action. Not all drugs use receptors. The example that is given in Lippincott is antacids, such as Tums. These are chemically going to neutralize excess stomach acid. But most drugs do use some type of receptor. And then toward the end of this lecture, we're also going to touch on a topic called pharmacodynamics, which deals with how the concentrations of these drugs affect how big or how small the effect of the drug is. So let's talk about the chemistry of receptors and their ligands. So when a receptor binds to its ligand, it does so via a chemical bond of some sort. Usually these are electrostatic bonds, such as ionic bonds. Sometimes they're hydrogen bonds. Sometimes they use the weak van der Waals forces. But these are not strong covalent bonds. These are much weaker bonds. And the distance between the atoms as they interact determines how strong that bond is going to be. So if the atoms can't get very close together, it's going to have a pretty weak bond. But if the shape of the receptor and the ligand is such that the atoms can get really close, it's going to be a much, much stronger bond. And the strength of the bond determines how selective the receptor is for its ligand. Usually when these receptors bind their ligands, it's a reversible binding, but sometimes it can be irreversible. And you have to have a good fit between the receptor and the ligand. That's what allows for specificity, so that epinephrine won't bind to an insulin receptor and insulin won't bind to a histamine receptor, and so on. You'll sometimes see simplified drawings where you have a nice round cup-shaped receptor and you have a nice round ball-shaped ligand, and they'll bind together very nicely. Or you'll have a triangular receptor and a triangular ligand. That's a simplified way of thinking about this, and that's what we call the lock and key model, where the key is the ligand and the lock is the receptor, and so it has to fit just right in order to unlock the door. That's sort of an old way of thinking about this. The more modern way is something we call the induced fit model, in which the receptor actually changes its conformation, it changes its structure and its shape in order to bind to the ligand. So the ligand comes along, and the receptor may be shaped like this, and because of the interaction of that ligand, the receptor opens up to fit that ligand very, very nicely. And that conformational change of the receptor is going to actually activate the receptor. And you'll see a nice animation of this in just a moment. We see drawings of the four major families of receptors in Figure 2.2. And we're going to talk about the basics of each of these types of receptors. I will give you some specific examples for each type of receptor, but the main focus here isn't on the specific examples, it's on the basic concept of how these receptors are going to work. So don't worry about memorizing a list of every possible drug that has a specific receptor type. Just get the idea of how the receptor types work. So the type of receptor may depend on the ligand. So if you have a hydrophobic ligand, it's going to be able to diffuse through the lipid bilayer cell membrane and get into the cell. So it can actually interact with intracellular receptors. But most drugs are more hydrophilic, and therefore they can't get through that cell membrane, so they have to bind to an extracellular receptor. So there are 4 types of receptors that we're going to talk about, 4 major families of receptors. They're the ligand-gated ion channels, the G-protein coupled receptors, the enzyme-linked receptors, and intracellular receptors. And we're going to talk about each one of these in turn. So the first type of receptor we're going to look at is ligand-gated ion channels, and this is on figure 2.2a. These are ion channels that regulate ion flow across the cell membrane. And the binding of the ligand may open or close the channel. And usually these channels have a fairly rapid response. It only lasts a few milliseconds. Some examples of this type of receptor would include the nicotinic receptor, in which acetylcholine comes in and opens up the channel and allows sodium to flow into the cell, and that generates an action potential. Another example would be GABA, or gamma-aminobutyric acid. Now GABA is an important inhibitory neurotransmitter. And when the presynaptic neuron releases GABA, that GABA goes across the synapse and binds to a receptor on the postsynaptic cell. And that receptor is a chloride channel that opens up, and it allows chloride ions to flow into the cell, and that hyperpolarizes the cell membrane, makes that postsynaptic cell less likely to fire, so it's an inhibitory transmitter. Drugs that are benzodiazepines, which include drugs like Valium and Xanax, which you've probably heard of, these drugs enhance the action of GABA. They increase the frequency of the ion channel opening that GABA produces, so there's increased hyperpolarization. So benzodiazepines increase the action of GABA on the postsynaptic cell. And then subdrugs act not on ligand-gated channels, but on voltage-gated channels. Drugs like this include lidocaine, which is a local anesthetic, and it acts on a voltage-gated sodium channel. The next family of receptors we'll talk about are very important. It's the G-protein coupled receptors. So the structure of these G-protein coupled receptors is a transmembrane portion, which is a single peptide with 7 transmembrane proteins that are represented like a bundle of sticks. So it looks like this big bundle of dynamite kind of stuck together, and it spans the cell membrane. And you have a portion of that sticks out into the extracellular space, and that's the part that the ligand binds to. And then within the membrane, you have this G protein, which is linked to that protein receptor. The G protein is made up of 3 subunits, alpha, beta, and gamma. The beta-gamma subunits are kind of joined together, and they usually stay within the membrane. Then there's an alpha component, which has an intracellular component, and that alpha subunit can bind to either GDP, or guanosine diphosphate, or it can bind to GTP, or guanosine triphosphate. So let's look at figure 2.3 and see how these G-protein coupled receptors function. So the ligand comes in and binds to the extracellular portion of the receptor, and that forces that transmembrane portion of the receptor to undergo a conformational change. That conformational change then activates the G protein so that GTP can come in and replace GDP on the alpha unit. And that causes the alpha subunit to undergo another conformational change, and it dissociates from the beta-gamma subunit. So both the beta-gamma subunit and the alpha subunit can go on and activate other enzymes. They can turn on other enzymes. And this signal is perpetuated downstream by what we call second messengers, which is whatever that activated enzyme is going to produce. And then those second messengers can go on and regulate other enzymes and so forth. Now, eventually, that G protein is going to self-hydrolyze the GTP back to GDP, and that sort of resets the molecule so that it's going to go back to its inactivated form and be linked back up to the original receptor peptide. So here we see an animation of the ligand binding to the receptor, and that causes a conformational change in the G protein, which then goes over and binds to another enzyme to produce its effects. The green substance in this animation is an enzyme called adenylyl cyclase, which produces cyclic AMP. So I mentioned that these G protein coupled receptors use a lot of second messengers, and we'll talk about the different second messengers here. One of the most common, most important ones is what we saw in the animation, adenylyl cyclase. It's an enzyme that turns ATP into cyclic AMP. And then cyclic AMP can go on and stimulate various other enzymes, especially protein kinases. And an example of a G protein coupled receptor that uses adenylyl cyclase is the histamine H2 receptor. Another important second messenger is called phospholipase C, and we see that in the histamine H1 receptor. The G protein activates phospholipase C, which then generates other second messengers, including diacylglycerol and inositol 1,4,5-triphosphate, which is also called triphosphoinositol. And then these two second messengers can regulate calcium levels, and they can also regulate other various proteins. Another second messenger system to know about is called guanylyl cyclase, which instead of converting ATP to cyclic AMP, it converts GTP to cyclic GMP. And then this can go on and stimulate protein kinases. The guanylyl cyclase system is really found in fewer cells because fewer cells use cyclic GMP, specifically the intestinal mucosa and vascular smooth muscle both use cyclic GMP as second messengers. So the second messengers can often activate other enzymes and propagate the signal, and so the signal is multiplied. So it's like the old thing where they tell two friends and then they tell two friends and so on and so on. The next type of receptor we'll talk about is an enzyme-linked receptor. So this receptor has enzymatic activity, and the ligand comes in and binds to an extracellular portion of the receptor, but intracellularly this receptor has some type of enzyme activity. Because this is an enzymatic reaction that we're catalyzing, the response is much slower than we saw with the ion channels. It lasts a lot longer than with the ion channels. It may last several minutes or it may last a few hours. Some examples of this include tyrosine kinase, which is a very, very common protein kinase that a lot of receptors will have tyrosine kinase activity. Insulin binds to a receptor that has a tyrosine kinase activity. A lot of different growth factors bind to receptors that have tyrosine kinase activity. And as we mentioned, the receptor molecule it undergoes a conformational change that activates the kinase. A kinase is an enzyme that phosphorylates a specific substrate. It could phosphorylate a protein or lipids or carbohydrates. It transfers phosphate from ATP to whatever the substrate is. In this case, it's a specific amino acid, tyrosine, so it's a tyrosine kinase. And then that added phosphate molecule modifies the 3D structure of whatever that target substrate is so that it acts as a molecular switch. It can either switch that protein on or switch that protein off. Very often these kinases will autophosphorylate. They'll actually phosphorylate themselves, so they'll switch themselves on. So the receptor makes that tyrosine kinase activity available, and then it phosphorylates itself, so it switches itself on. And that will propagate the signal as well. The fourth family of receptors are the intracellular receptors. So this is very different. In this situation, the ligand has to be hydrophobic enough to diffuse through the cell membrane and get into the cytosol where the receptors are housed. An example of this includes steroid hormones such as cortisol or estrogen or testosterone. The ligand, whether it's the hormone or some drug acting on that same receptor, is going to enter the cell, bind to the receptor inside the cytoplasm, and then that ligand receptor complex usually will move into the nucleus of the cell where it can bind to specific DNA sequences and it regulates gene expression. So because we're regulating gene expression, we're regulating protein synthesis, this has a much slower onset of action. It generally takes more than 30 minutes to have its action, and it has a longer duration of action, sometimes several hours, sometimes it can last days. Because it's affecting gene transcription, it's just going to last a lot longer. So let's talk about the concept of spare receptors and amplified signals. We already saw how receptors can multiply or amplify a signal through second messengers. It just takes one receptor-ligand complex to activate more than one messenger. So that protein receptor may activate several G proteins. Also, activated messengers, such as our G protein, can last a lot longer than the original receptor-ligand complex does. And then the second messengers, in this case we talked about cyclic AMP being a common second messenger in G protein systems. That second messenger, or cyclic AMP, may exert their effects a lot longer than the original G protein lasts. So because of this, you only need a small fraction of the receptors to be activated in order to get maximum cellular effects downstream. All the other receptors that aren't being activated are called spare receptors. So, for example, it's estimated that about 99% of the insulin receptors in the body are spare, meaning you don't have to activate them to get full effect of insulin. It gives us a huge functional reserve in order to make sure that there are plenty of opportunities to get adequate glucose into the cell, which is the role of insulin. Even if half of the cell's insulin receptors are knocked out or dysfunctional for some reason, you still have plenty of insulin receptors that are active and available to bind to insulin. But take an example of beta adrenergic receptors in the heart. It's estimated that only 5 to 10% of these receptors are spare. So in order to achieve maximum contractility, you're going to have to activate the majority of the receptors that are available. And that leads to a very low functional reserve. It means if you give a lot of a beta agonist in a heart failure patient, you're not going to get a huge increase in contractility. There's only so many spare receptors that are available to that beta agonist that you're giving. Next, we need to talk about desensitization of receptors. Now, repeated signaling or continuous signaling can change a receptor's response. Sometimes if you overstimulate a receptor, it can lead to damage to the cell, or it can actually cause cell death. For instance, electrolyte imbalances, if you get a lot of calcium flowing into a cell over and over and over again, that high calcium level will trigger cell death. So the cells have built-in mechanisms to protect themselves and minimize the effects of this overstimulation. One of these is called tachyphylaxis, and that's where a receptor becomes desensitized to the action of the ligand. Repeated stimulation leads to a diminished response, a lower amplitude response, as you see in the figure. If you wait a little while, though, the receptor becomes sensitized again. The sensitivity goes back up to normal. So when you wait a while and then you stimulate the receptor again, you're going to get the same response as you did before. Another mode of desensitization is called downregulation of receptors. Sometimes membrane-bound receptors will actually undergo endocytosis and be taken into the cell and taken out of commission. They're no longer extracellular at all, so there's no way for the ligand that's out there, the drug or whatever it is, to bind to those receptors because the receptors have been taken out. And then some receptors, especially voltage-gated channels, show what we call a refractory period. It means that they require a rest period of a definite amount of time before they can be activated again. That may be 100 milliseconds or something, but it allows for the cell to rest and recuperate from the action of that receptor before it's going to be stimulated again.
Now let's pause for a moment and review the material that was just covered. Before we do that, if you haven't already done so, this might be a good time to read your pharmacology text that corresponds to the material that was just discussed. Then together we'll go through the quick review in the study guide.
What neurotransmitter opens ligand-gated channels so that chloride enters and hyperpolarizes the cell? GABA? Oh, that's right! GABA is the main inhibitory neurotransmitter in the brain, while glycine is the main inhibitory transmitter in the spinal cord. There are two kinds of GABA, GABA A receptors and GABA B receptors. It is GABA A receptors that are ion channels which increase chloride influx and hyperpolarize the cell. Also, benzodiazepines such as Valium and Xanax act on these GABA A receptors and potentiate the inhibitory effects of GABA. That's such a fun word. GABA, GABA, GABA.
Hi, I'm Dr. Chris Lewis. I'm here for your quick review number 1. Let's get started. Do most receptors bind to their ligands reversibly or irreversibly? The answer, reversibly. To which family of receptors do the following ligands bind? First one, nicotine. That's ligand-gated ion channel. Estrogen, intracellular receptors. Insulin, enzyme-linked receptors. Histamine, G-protein-linked receptors. lidocaine, voltage-gated ion channel, GABA, ligand-gated ion channel, cortisol, intracellular receptors, and testosterone, intracellular receptors. Which subunit of G proteins binds GTP? The answer, alpha subunit. What kind of reaction is catalyzed by tyrosine kinase? Kinases catalyze the transfer of phosphate from ATP to a substrate. This is called phosphorylation. In the case of tyrosine kinase, the substrate is the amino acid tyrosine. What do second messengers, namely inositol 1,4,5-triphosphate and diacylglycerol, do? Well, they regulate intracellular calcium, and they transfer signals from hormones, neurotransmitters, and drugs. What type of channel will regulate the flow of ions across the cell membrane? That's the ligand-gated ion channel. List 3 ways that G protein-linked receptors amplify signal duration and intensity. Well, first, a single ligand receptor complex can interact with many G proteins. Second, activated G proteins persist longer than the original ligand receptor complex. And then finally, second messengers can exert their effects longer than the original G protein. What is the name of the process where cells endocytose receptors in order to protect themselves from overstimulation? And we call this downregulation. That concludes quick review number 1. Now back to the lecture.
So that pretty well covers ligands and receptors. Next, we're going to talk about pharmacodynamics, which goes into how the concentrations of these drugs affect the magnitude of the response you're going to get from them. So the big topic for pharmacodynamics is what we call a dose-response relationship, and this is on page 30. There are 2 main types of dose-response relationships we'll discuss, a graded dose response and a quantal dose response. So let's talk about graded dose response relationships first. Basically, this means that if you give more drug, you get more response. It's a continuous, gradual slope. Imagine we're talking about blood pressure drugs, and we're going to talk about blood pressure drugs throughout several sections of this. If you give a small dose of a blood pressure drug, you might expect a small drop in the blood pressure. If you give a larger dose, you can expect the blood pressure to fall lower. That is a graded dose response. In contrast, we'll talk about a quantal dose response, which is an all or nothing response. So on page 30, figure 2.6a, it shows this graded dose response curve. On the x-axis, you have the concentration of the drug, and as you increase concentration, you get more of a response until eventually, out there toward the right part of the graph, you're maximizing the response. This curve is called a rectangular hyperbola, and it's very important in biology. We see it in a lot of different circumstances. We see it here in ligand binding, we see it in enzymatic reactions, enzymatic activity, and we're going to look at this hyperbolic curve again in just a little bit. From this curve, we can determine 2 important properties of the drugs we're talking about. We can determine potency and efficacy. And these are really important both in clinical practice, and they're very important for your boards as well. So this is a very high yield topic. We're going to talk about potency and efficacy in turn. So potency describes how much of a drug is needed to give an effect of a given magnitude. So again, if we're talking about blood pressure drugs, we might say how much blood pressure drug is needed to lower the blood pressure by 20 points. And the potency is determined by the amount of drug required to achieve 50% of whatever the maximum effect could be. So if the maximum amount of blood pressure lowering you're going to get from a given drug is 20 points, we're looking at how much drug it takes to lower it by 10 points, or 50% of that maximum amount. And we call this amount of drug EC50. That stands for the effective concentration to give us a 50% response, or the half maximal effective concentration. So EC50 is an amount. It's a dose of a drug. It's a concentration of drug. You see that the x-axis is the concentration of the drug required, and the y-axis is the percentage of the maximum effect. So we see 2 drugs, drug A and drug B, and they have the same maximum effect. They both lower the blood pressure by the same amount. Maybe that amount is 20 points, 20 millimeters mercury. But the 2 drugs require a different amount of the drug in order to achieve that effect. They have different potencies. So look at the graph. We arbitrarily mark 50% of the maximum effect, and we follow that horizontally out to where it intersects the curve. And the value of x the value of the x-axis at that point where it intersects the curve is the EC50. So you see for drug A, the EC50, well, let's say it's 4 milligrams, but the EC50 for drug B, to achieve that same maximum effect, to achieve 50% of that maximum effect, the EC50 is much higher. Say it's 40 milligrams. So in order for drug A to lower the blood pressure by 10 points, or half maximum effect, you only have to give 25 milligrams. But in order to get that same effect and lower the blood pressure 10 points using drug B, you have to give more. You have to give 60 milligrams. So which drug is more potent? Well, I haven't really told you exactly, but you can probably figure it out intuitively that drug A is more potent. It takes less of drug A, it takes a lower dose of drug A to get that same effect. So drug A is more potent. It has a higher potency. So Lippincott gives us the example of a drug class called angiotensin receptor blockers, or ARBs. And the two drugs they talk about are candesartan and irbesartan. And they're both blood pressure drugs. That's convenient. Candesartan is more potent than irbesartan. Candesartan is given in small doses, 8 milligrams, 16 milligrams, 32 milligrams. Irbesartan is given in much higher doses, 75 milligrams, 150, 300 milligrams. So both drugs can achieve the same drop of blood pressure, but it takes a lot more of irbesartan to do it. So irbesartan is less potent than candesartan. Now, in the real world, there might be several orders of magnitude involved in this. It might not be just from 8 milligrams to 32 milligrams. It might be from 8 milligrams to 800 or 8,000 milligrams. So it might be a huge difference in the orders of magnitude involved and the concentrations of the drug involved. So we don't plot this on a linear scale. We plot it on a logarithmic scale. So as you see here, the x-axis is plotted with a logarithmic scale. So it goes from 0.1 to 1 to 10 to 100 and so on. So instead of having a curve that's a rectangular hyperbola, you have this S-shaped or sigmoid curve. So the other important drug property that these dose-response curves can demonstrate is efficacy. Now, efficacy is the ability of a drug to elicit a physiologic response when it interacts with a receptor. So it answers the question, how strong a response do you get from that drug? We're talking about blood pressure drugs. The question is, how much is that drug going to be able to lower the blood pressure? Is it going to lower it 20 points, or is it only going to be able to lower it 10 points? Efficacy depends on a couple of different factors. It depends on how many drug receptor complexes are formed, and it depends on how efficiently those drug receptor complexes can actually elicit a biological response, a cellular response. From the dose-response curves we've been looking at, instead of comparing the EC50, as we did with potency, we're comparing what the maximum biologic effect is. We're looking at where does that curve plateau, where does it peak out? Are you able to lower the blood pressure by 20 points, or are you only able to lower it by 5 points or 10 points? So when you look at figure 2.7 on page 31, you can see that drug C has a much lower biological effect than drug A or drug B. No matter how much drug C you give, you've already plateaued. You're never going to be able to get up to the same level of effect as drug A or drug B. Drug C has an inherently lower efficacy than the other two drugs do. Its intrinsic activity is just lower. So that covers potency and efficacy, and these are two really important concepts. But then we turn to page 31, and you see all these formulas, and honestly, it gives me a headache just looking at them. The good news is this is not a terribly important formula. You're not going to have to do calculations based on this formula. But it's important to understand the relationship between these different variables. So we're talking about drug receptor binding. And you look at that first equation, and let me just define a couple of the parameters here. First of all, Kd is called the dissociation constant of the drug from the receptor. It determines how much affinity the ligand has for the receptor. So a high dissociation constant, a high Kd, means a weaker interaction between the drug and the receptor, and it has a lower affinity. If you have a low Kd, that means it's a higher affinity. The receptor is going to bind really tightly to that ligand. So look at the right side of the equation there. We have D over Kd plus D. So D is your concentration of the drug, And at higher and higher doses of the drug, when the drug concentration D is much, much higher than KD, KD is pretty much negligible. If D is 1,000,000 and KD is 1, then 1,000,000 divided by 1,000,001 is really just, it reduces to 1, or it approaches the value of 1. So at a high drug concentration, this equation equals 1. So then look at the left side of equation. If we're talking about the ratio between the drug receptor complexes, which is DR, to the total receptors available, which is R sub T. So at a value of 1, when you have a high concentration of drug and the equation equals 1, the total number of drug receptor complexes is equal to the total number of receptors, which means all of the receptors are bound to the drug. So this is a maximum effect. When all the receptors are bound, that is going to have the maximum biologic effect. So then we look at the relationship of binding to effect. Equation 2 down there at the bottom of the page says basically the same thing. The right side of the equation is the same. At high drug doses, the equation is still going to approach a value of 1. And the effect achieved by that is E. And the effect achieved is basically equal to the maximum effect, or E max. So I'm not really sure why they went into this, because both of these equations have the same graphs we've already been looking at. When you look at figure 2.8, it's the same types of curves we've already been looking at, but it's just using different values for the x-axis and the y-axis. So next we need to talk about agonists and antagonists. And this is another very important concept. So an agonist is a substance that binds to a receptor and elicits a biologic response. For drugs, we're usually talking about a drug that's going to mimic the effect of an endogenous ligand. For example, we have a drug called phenylephrine, which is an agonist at the alpha-1 adrenergic receptors. We also have a drug called albuterol, which is an agonist of the beta adrenergic receptors. These bind to the same receptors as the endogenous ligand epinephrine, and they elicit the same response as epinephrine. Sometimes we talk about drugs being full agonists, which means they elicit the same magnitude of response, and we'll talk a little bit later about a partial agonist, which elicits a much lower response. And then we'll talk about antagonists. An antagonist is any substance that reduces or diminishes the receptor-mediated response. So again, for drugs, we're usually talking about a drug that is going to reduce the action of another drug or reduce the action of an endogenous ligand. It's going to oppose the action of that ligand, and it's going to prevent it from binding to the receptor. Usually antagonists have a pretty high affinity for their receptors. So we talk about competitive antagonists and non-competitive antagonists. The competitive antagonist is going to bind to the same site of the receptor as the endogenous ligand, or as the agonist does. So it's competing with that agonist for the receptor site. So that's why we call it a competitive antagonist. It's competing for the receptor site. So if we're going to stick with our blood pressure example, there's a drug called prazosin, which is an alpha receptor antagonist. It's going to compete with epinephrine at those alpha adrenergic receptors. Then we'll talk about non-competitive antagonists. This is a little bit different. It's not going to compete for that same receptor site. Non-competitive antagonists bind to the receptor at another site, which we call an allosteric site, and it is going to inactivate that receptor, so that receptor cannot bind with the agonist. It's not interfering with the agonist receptor site. It's interfering with a different site. Sometimes drugs will act as chemical antagonists, meaning it's not really binding the receptor, but it's binding to the drug. It's binding to the agonist itself and taking the agonist out of commission. And sometimes we'll talk about drugs being functional antagonists or physiologic antagonists. This means that the drug's action opposes the action of the agonist, but it doesn't really use the same receptor to do that. It's not really an antagonist in the classic sense, but the end result is the same. So the example in the book is epinephrine as a physiologic antagonist of histamine. For example, if you've got a patient who's having an allergic reaction, they're having a lot of histamine circulating, and that histamine is causing bronchospasm. Well, you may know, what do we give for an allergic reaction? We give epinephrine. because that's going to reverse the bronchospasm. But it doesn't act on the histamine receptors. If you wanted to act on the histamine receptors, you'd give an antihistamine. But instead we give epinephrine, which acts on the beta receptors, the beta adrenergic receptors, and that's going to reverse the bronchospasm. So it's antagonizing the effect of histamine, but it's not antagonizing the histamine receptor whatsoever. This is called a functional antagonist or a physiologic antagonist. So here we see a curve that shows the effect of a competitive antagonist. Here in red, we have the curve of an agonist by itself. It could be an endogenous agonist, like epinephrine is an agonist at adrenergic receptors, or it could be an agonist drug, like albuterol, which is an agonist at the beta adrenergic receptors. And then here the orange curve shows an antagonist given with a competitive antagonist. So to use our example, we'll use the beta adrenergic antagonist called propranolol. So the propranolol competes for the receptor binding sites so that for any given concentration of agonist, the biologic effect is reduced. So if at this dose of epinephrine you got this amount of response, when you add in the antagonist, you get a much lower response. So how do you overcome the antagonism? By increasing the amount of agonist. If you start with 5 agonist molecules and 5 antagonist molecules that are both competing for receptors, and then you just added enough agonists, so instead of 5 agonist molecules, you had 100 agonist molecules, and they're still competing with just 5 antagonist molecules, you essentially overwhelm the antagonist. And you can see from the curve that even with the antagonist, if you increase the dose of agonist given, you're eventually going to get to the same efficacy or the same maximum response even with the antagonist. You get the same 100% biologic effect. It just takes a lot more agonist to do so. So to use the concepts of potency and efficacy, the competitive antagonist has reduced potency but not efficacy. But this is only true for competitive antagonists. So now we see a curve for a non-competitive antagonist. And again, in red, we have the same curve for the agonist. And in orange, we have the agonist given with a non-competitive antagonist. So here the antagonist isn't competing for receptor sites. It's binding to the receptor at another site and changing the conformation of the receptor molecule so the agonist cannot bind. But you see, it doesn't matter how much agonist you give. You can give tons of agonist and you're still not going to get up to the same effect as you would with the agonist by itself. The curve plateaus at a much lower maximum effect. You could have 1,000 agonist molecules, but if all the receptors have been taken out of commission, no amount of agonist will do anything about that. The effect of the non-competitive antagonist is to reduce efficacy, but it doesn't affect potency. And you can see that the potency of these two curves is exactly the same. EC50 hasn't changed. EC50 for the agonist by itself is right here at somewhere between 1 and 10, we'll call it 4. EC50 for the agonist plus the non-competitive antagonist, the orange curve, is right there. It's at the same place. So it doesn't affect potency at all. It only affects efficacy. All right, so next I want to go over these same curves with some different terminology that you may run across on your boards. We're talking about Km and Vmax, and these are very high-yield topics for your boards. This is also covered in Chapter 1 on pharmacokinetics when we're talking about drug metabolism, but it applies here as well. And I'll just briefly review what we covered in Chapter 1. So here we have Km, which is the Michaelis constant. Km reflects the substrate concentration or the drug concentration. So Km is a concentration, it's an amount of drug. Km is the concentration of substrate that's required to get the reaction velocity to half of Vmax. So in other words, Km is the concentration that you achieve half Vmax at. Km is basically equivalent to EC50, which is the concentration required to get half of the maximal response. This is just a different way of plotting the same data. This is called a Lineweaver-Burke plot, named after two chemists, Dr. Lineweaver and Dr. Burke. So the slope of the plot is Km divided by Vmax. Where the plot intersects the x-axis is 1 over minus Km. And where the plot intersects the y-axis is 1 over Vmax. So you need to be able to recognize what those intersections mean. If you increase the y-intercept you have a lower Vmax because these are inversely proportional. If the x-intercept moves to the right, it means you have a higher Km, which means a higher concentration of drug is required to reach half Vmax or to reach EC50. It means it's a less potent drug. So let's look at a Lineweaver-Burke plot with antagonists. On this Lineweaver-Burke plot, the yellow line is the uninhibited agonist, which is the normal value or the baseline value. When you add a competitive inhibitor, which is this graph in orange, you're not changing the y-intercept, you're not changing Vmax, you're not changing the maximum response, you're not changing efficacy with a competitive inhibitor. What's changing is the x-intercept is shifting over to the right, so you're changing Km or EC50. You're going to need more of the agonist to maximize the biological effect. And the pink graph is a non-competitive inhibitor that's added, so you're not changing Km, the x-intercept is the same. What you're changing is the y-intercept. It's higher, so you have a lower Vmax or a lower maximum effect. You have lower efficacy. You really need to know this stuff for the boards. We've had students tell us you need to know Km and Vmax like the back of your hand. This is very, very high yield. Okay, so let's go back to Lippincott for just a couple of quick things. We already briefly mentioned the idea of a partial agonist. A partial agonist has a lower efficacy than a full agonist. The maximum response is lower. The efficacy is lower. This is independent of the affinity of the ligand for the receptor. A partial agonist ligand may have higher or lower affinity for the receptor compared to the full agonist. It's just a different efficacy. It's not going to give you the full response like a full agonist would. So a partial agonist basically competes for the receptor binding site just like a competitive antagonist would. So if you have a large concentration of the partial agonist and a relatively small concentration of the full agonist, the partial agonist is going to win out. It's going to win that competition, and you're going to get an agonist response that's blunted or reduced compared to what you would with the full agonist because you're getting the effect of the partial agonist. That's what we see in Lippincott, page 2, or rather figure 210. We see that if you add enough partial agonist, you reduce the agonist effect to the level of the maximum effect of the partial agonist. And they give you the example of a neuroleptic drug called aripiprazole. That's a drug that's used for psychotic disorders such as schizophrenia. So aripiprazole has partial agonist activity at certain dopaminergic receptors. So it's going to stimulate pathways that aren't getting enough dopamine. So if you've got a pathway that needs dopamine, it's not getting enough, you can give a partial agonist and it's going to give you some of that dopaminergic transmission. But aripiprazole will also inhibit pathways that are getting too much dopamine because it's going to reduce the amount of response to that dopamine because you're getting a partial agonist effect. And it's going to compete with the endogenous dopamine at those receptor sites. So the last topic we'll talk about is this concept of the quantal dose-response relationships. We've been talking about graded dose-response curves in which the magnitude of the response depends on the dose. But now we're going to shift gears and talk about quantal dose-response curves. Quantal means all or nothing. So does an individual respond at a certain dose or not? Generally, we're talking about big populations rather than individuals with this. So what proportion of the population is going to respond at any given dose? All the population, half the population, a third of the population? And you can also quantize those dose-response curves. You can say, does a given blood pressure drug drop the blood pressure by a necessary amount, a minimum amount? If you set that threshold at 5 milligrams, you can say, yes, at this dose, this drug drops the blood pressure by 5 milligrams or 5 millimeters mercury, or it doesn't. It's all or nothing. So we can plot what percentage of the population responds by that set amount at a given dose. So if you look at figure 2.11 on page 33, and just ignore all the details and look at the axes and look at a single curve, you can see that the x-axis is the dose plotted on a logarithmic scale. So we have that sigmoid curve, that S-shaped curve. And the y-axis is the percentage of patients who respond. And we use this to demonstrate the concept of therapeutic index. So therapeutic index is a ratio of the toxic dose to the therapeutic dose. By convention, we don't worry about what is toxic to 100% of the population or what is therapeutic to 100% of the population. We worry about what is toxic to 50% of the population for these calculations and what is therapeutic for 50% of the population. So the formula there on page 33 is therapeutic index is equal to TD50, or the dose that's toxic to 50% of the population, divided by ED50, or the dose that's effective for 50% of the population. And this is one of those high-yield concepts you really need to know and understand. You're going to see this again on your boards. Sometimes instead of toxic dose, you might see lethal dose, or LD50. So therapeutic index is a measure of the drug's safety. The higher the therapeutic index, the more safe a drug is. Think about it, because this may not be completely intuitive. If you have a high therapeutic index, TD50, the toxic dose, is going to be much higher than ED50. That means that most people, or at least 50% of people, will have a therapeutic effect at a much lower dose than the toxic dose. And for most people, you'd expect to have to give a dose much higher than the therapeutic dose in order to get a toxic effect. So some drugs are talked about as having a very large or very wide therapeutic index. The example given is penicillin, and you see that on figure 2.11b on page 33. There's a big difference between those curves. They're widely separated. So you can give about 10 times more than the therapeutic dose of penicillin and still not get into toxicity. It has a very wide or very large therapeutic index. Some drugs, on the other hand, have a very small or very narrow therapeutic index. The example in Lippincott is warfarin, but other drugs that have very narrow therapeutic index include lithium, digoxin, phenobarbital. And what do these drugs have in common? These are all drugs that you have to monitor drug levels on. So you have to monitor this closely because there's a very narrow therapeutic index. If you look at figure 2.11a, you can see that there's an overlap between those two curves. They're not separated very much. They're closer together. And so that means that the dose required to get a therapeutic response for some patients, kind of at the upper end of that curve, of the therapeutic curve, is going to be toxic to some patients. So you may have to give enough drug to be therapeutic, but that may be enough to cause toxicity. So that drug has a very narrow therapeutic window, a very narrow therapeutic index.
Now let's pause for a moment and review the material that was just covered. Before we do that, if you haven't already done so, this might be a good time to read your pharmacology text that corresponds to the material that was just discussed. Then together we'll go through the quick review in the study guide.
What do we call it when a receptor becomes less responsive to a drug due to continuous or repeated administration? What kind of a student are you? Well, if you don't know it, look it up. Do you think I'm going to spoon-feed you your entire medical education? Take some initiative. It's all right here. Desensitization. Desensitization of receptors. Ugh, I can't even look at you.
All right, we're back for our quick review number 2. Let's get started. What is EC50? Well, EC50 is the concentration of drug that produces an effect that is 50% of the maximum. Next, drugs A and B are both antihypertensive drugs. Drug A has an EC50 of 180 milligrams, while drug B has an EC50 of 100 milligrams. Which drug is more potent? Well, the answer is drug B. The lower the EC50 will indicate a more potent drug. Next, complete the following table. Will higher doses of the agonist overcome the inhibition? And we're comparing this to competitive antagonists and non-competitive antagonists. So for this one, the competitive antagonist, the answer is yes. For the non-competitive antagonist, the answer is no. Does the antagonist bind the receptor at the same site as the agonist? For competitive, the answer is yes. For non-competitive, the answer is no. What effect does the antagonist have on the efficacy of the agonist? For competitive antagonist, no effect. For the non-competitive antagonist, it's reduced. What effect does the antagonist have on potency of the agonist? For competitive antagonist, the answer is reduced. For non-competitive antagonist, the answer is no effect. Label the following plots as either competitive or non-competitive inhibition. Well, our chart on the left here, you see our top curve and our bottom curve. So the top curve will be A, a competitive, and the bottom one will be B, non-competitive. On the right side, with our three lines. The very top line will be A, non-competitive, and the bottom line will be B, competitive. What is the formula for therapeutic index? Well, therapeutic index will equal TD50 divided by ED50. So you've got your toxic dose divided by your effective dose. Now, sometimes you'll see LD interchanged for TD, and that'll be lethal dose as opposed to toxic dose. Drugs that oppose the action of the agonist but do not use the same receptor are called what? These are the functional antagonists. Next, drugs that bind to the allosteric site are called what? And these are also known as our non-competitive antagonists. And that's going to conclude our quick review number 2. Now's a good time to pause the video and complete the in-session quiz. Then restart the video and we'll go through the quiz together. Alright, it's time for our in-session quiz. Let's get started. Put the following steps of G protein signaling in chronological order. Well, the first thing that happens is unoccupied receptor does not interact with GS protein. The next in order is occupied receptor changes shape and interacts with GS, which releases GDP and binds GTP. Next is alpha subunit of GS protein dissociates and activates adenylyl cyclase. And then finally, GTP on the alpha subunit is hydrolyzed to GDP and adenylyl cyclase is deactivated. Next, why is cortisol able to bind to intracellular receptors? Well, cortisol is hydrophobic. Another way of describing it would be lipophilic, so it loves lipids. So this can now diffuse through cell membranes, which is a hydrophobic lipophilic entity. So it can interact then with the intracellular receptors in the cytosol. Define potency and efficacy. Well, potency is a measure of the amount of drug needed to produce an effect of a given magnitude. So what dose is required to achieve 50% of the maximal biologic effect? Efficacy is the ability of a drug to elicit a physiological response. So in other words, what is the magnitude of that maximal biological effect? Next. Drugs X and Y are both antihypertensive drugs capable of lowering the systolic blood pressure 20 millimeters of mercury. Drug X has an EC50 of 50 milligrams, while drug Y has an EC50 of 100 milligrams. How much would you expect a 50 milligram dose of drug X to lower a patient's systolic blood pressure? So 50 milligrams of drug X should produce a half maximal response, lowering the systolic blood pressure by 10 millimeters of mercury. Which drug is more potent? Well, drug X has a lower EC50, and this indicates a higher potency. Drug Z is capable of lowering the systolic blood pressure 25 millimeters of mercury. Compared to drug X, does drug Z have greater potency or greater efficacy or both? Well, drug Z has greater efficacy than drug X, but you can't compare potency in the drugs with different efficacies. What is the difference between a competitive antagonist and a non-competitive antagonist? Well, the competitive antagonist will compete with the agonist for the receptor's binding site at that actual site, and this will affect the potency of the agonists. Now, a non-competitive antagonist will bind to an allosteric or other site, and this will inactivate the receptor itself, so this will lower the efficacy of the agonist. Next, does a non-competitive antagonist affect the potency of the agonist, the efficacy, or both? Non-competitive antagonists affect the efficacy of the agonist, but not the potency. Label points A and B. Well, A is going to be 1 over negative Km, and B is going to be 1 over Vmax. Next, would you expect a very safe drug to have a low therapeutic index or a high therapeutic index? Well, you're going to expect a high therapeutic index, or a broad or a wide therapeutic index is another way of describing it, and this will indicate greater safety. List 4 drugs with low therapeutic index. So that means that their toxicity dose is very close to their effective dose. Now, the 4 medicines that we talked about in this lecture are warfarin, lithium, phenobarbital, and digoxin. So these are risky medications. And what do all 4 of these have in common? Well, we all measure levels in these medications to make sure you don't get to that toxicity dose. How does a partial agonist work in comparison to a full agonist? Well, a partial agonist is less efficacious in general than the full agonist, but it can also have the same, less, or more affinity, or in other words, the ability to bind to that receptor than the full agonist. So the partial agonist can act as an antagonist to the full agonist in some settings. So a good example, and it was also mentioned during the lecture, is aripiprazole. So aripiprazole is an atypical neuroleptic, and it has partial dopamine activity. So in general, you're going to get less dopamine overall because it's blocking those dopamine receptors. But since it's stimulating a little bit of dopamine release, what's one of the benefits of aripiprazole is that you don't get some of the side effects that comes along with a full dopamine blockade. So that little bit of stimulation can be beneficial, though the overall effect is still as an antagonist. And that's going to conclude your in-session quiz.